Alright, so last chapter for this unit, chapter 5. We kind of was talking before I started recording. Decided the game plan is I'm going to lecture as long as I can today. Try and get through as much of this lecture. Finish up whatever we have left on Thursday, and then just do problems. Hopefully test still Tuesday, but there is a possibility I may push it to Thursday, right? I will definitely know by Thursday. If not before then, if I don't post an announcement for you guys. And we'll just work around it, Amber, no matter what I do. You're welcome. Alright, so chapter 5, I was so excited. This one is the one that has one of the things I started to talk about ahead of time. The calico cats, right? If you read ahead, right, this is the one with the calico cats. So, and I posted a link on our page, and I'll go to that if we get to that, which we should. So for this chapter, we have to understand things, right, to be able to predict what offspring are going to look like based on their genotype, right, what their phenotype is going to be. So we've learned several different things like dominant versus recessive. We've looked at gene interactions. We've even looked at how sex relates, and even environment to a certain extent. So Mendelian inheritance patterns are ones that directly influence the outcome of the organism's result, right? So most of the pea plant stuff that we looked at. And obey the laws. So what are the two laws that we need to know? Go ahead. Right, one's the law of segregation. What's the other one? Independent assortment. So in general terms, anyone want to take a stab at what we mean by independent assortment? Describe it in your own words. It doesn't have to be a definition from your book, right? Because you need to have a working understanding of these laws. Think about it in terms of chromosomes. Right? So how many chromosomes do we have? 46, right? 23 pairs, right? Where do they come from? Mom and dad, right? So you get one set from mom of 23, one set from dad of 23, right? Now when you make your sex cells to give to your potential babies, do you give them all your mom's chromosomes or all your dad's? No, it's a random mix, right? That's the independent, those chromosomes segregate independently, right? Still with me? Independent assortment applies when we start looking at more than one trait, right? So say something that's on chromosome 4 is not affected by something that's on chromosome 15, right? Those are going to sort independently. Unlike, what's a phenomenon that we looked at where stuff doesn't sort independently? One goes with the other. It's quote-unquote linked. Excellent. Right? That's where independent assortment doesn't apply, right? Because what's on the X chromosome goes with the X chromosome. You can't separate those two traits. Does that make sense? Where if we looked at two different traits, like say the shape of the pea and the color of the pea, those happen to be on separate chromosomes. So those sort independently. We have to look at them as each individual event. Still with me? Okay, right? So we need to have working definitions and understanding of Mendel's laws, right? Because we build upon that as we look at all these deviations. So most of them follow that inheritance pattern. However, though, there are many that do not, and that's what we've been looking at, right? And are going to continue to look at. So in this chapter, we'll discuss additional, even sometimes referred to as bizarre, patterns of inheritance that deviate from that Mendel pattern, right? Where they don't segregate or they don't independently assort. And so we're going to look at maternal effect and epigenetic inheritance. And these involve genes in the nucleus, right? So, and really, truly, up to this point, we've only been talking about DNA found in the nucleus. Is that the only place DNA is found in a cell? No, where else is there DNA? In our cells. In our mitochondria. Okay. We refer to that as extranuclear, or we sometimes specifically say mitochondrial DNA. Now with plants, they've got one more organelle that also has DNA. What organelle is that? Do plants and algae have? That allows them to photosynthesize, chloroplasts, right? Chloroplasts have their own DNA. So the genotypes of the offspring does not directly govern the phenotype as predicted with Mendel for these nuclear traits that are affected by the maternal effect in epigenetic inheritance. And then the other half of the chapter talks about extranuclear inheritance. So this is the mitochondrial and the chloroplasts. And also we'll talk about, how that's inherited. And of course it's different than what we expect. I'm on Linnell and Angela, right? I'll mark you guys because I have it actually open on the other computer because I was waiting on this one. I'm going to get it done now. Well, I have it staring at me and then I'm going to close it down because it's bugging me. Okay, so the first thing we need to do is be able to, of course, define what we mean by maternal effect, right? What is this maternal effect? And then be able to predict outcome of crosses from genes that exhibit maternal effect pattern of inheritance. And I think I am going to kind of narrow down the study guide for you guys, too. So, depending on how quickly I can get that done. I'll give you an idea, because a lot of these all have, predict crosses, and, I don't want you guys doing a gazillion crosses on the test. I'm going to limit it to a certain number, and I'm going to pick them and let you know which ones ahead of time. So by Thursday will be my goal for that. And then, of course, be able to explain at the molecular level this pattern of maternal effect. So what is maternal effect? This is an inheritance pattern with certain nuclear genes. So again, we're still talking about ones in the nucleus, in which the genotype of the mother directly determines the phenotype of the offspring. So it doesn't matter what the offspring have or end up having, or even what dad contributes. It's all about what the mom's genotype is going to determine the phenotype of those children. And the reason for this, and this is especially for organisms that nurture, you could say, their young, because mom gets prepared. And this makes sense too when we talk about the extranuclear stuff. I'll give you guys a little clue ahead of time. You get your mitochondria, and I probably have already said it, you get your mitochondria from your mom, right? Is your mom makes all that stuff, that egg for you, right? With the mitochondria and the rough endoplasmic reticulum and all the ribosomes and everything. All the sperm is a mobile DNA. So he's got a few little mitochondria to spin that good old flagella. But other than that, that's it. And some enzymes to penetrate through to get to that egg. But all your organelles and everything come from your mom. And a woman's egg is actually the largest cell in their body. Just like an ostrich egg is actually a single cell, right? It just has a whole lot of nutrients in that cell, right? Because that baby's got to have everything it needs for the first, I don't know how many days, inside that egg. Unlike us, right, we have the placenta, we can continually give nutrients, but there's even a phase where, they are going to have nutrients, right? They're going to have the yolk sac, and they're going to have to provide, what they need from that until the placenta fully develops. So all that stuff is already there. So think about that too. She makes stuff for you, right? She makes a whole bunch of ribosomes, right? May make a whole bunch of enzymes that you're going to need ahead of time. So bam, once that sperm gets there, we're ready to go, right? We've got everything we need, and those cells divide very rapidly. because they really don't have to copy a lot of that stuff. It's already been made. They're just divvying it up. Does that make sense to you guys? So that's why this kind of makes sense. It's like, oh, okay, well, mom prepared this stuff for the baby. That's why it's the mom's genotype that determines it, because she made it ahead of time for the babies. This is chapter 5. Yeah, it should have been. I posted a whole bunch of stuff. Was it not? Are you guys not seeing it? Under modules, under objectives, and or PowerPoints or whatever. Where I've been posting everything. Yeah, you might be. I'm not going to stop class to help you, Angela. Okay, so the genotypes of the father, as I said, and the offspring themselves do not affect the phenotype of the offspring. And again, this is an accumulation because mom made all this stuff ahead of time, right? So that's why what the baby is doesn't factor in. What dad is doesn't factor in because he hasn't arrived yet on the scene, right? Mom made this ahead of time for the baby. So that's why it's her genotype and her genotype only that determines the phenotype of this particular characteristic that has maternal effect. So the example that they give was discovered by Boycott in the 1920s, and he was working with water snails. And he found that the shell of the snails had a certain rotation to it, whether it be right-handed or what's referred to as left-handed. So, or dextral or central, or central. Yeah, that one's a tongue twister. And so dextral was more common, right? So he surmised that that's probably the dominant trait out of the two. So the snails, that curvature, right, to the shell and the whole body plan within the shell is based on the cleavage pattern as those cells divide, right? When it goes from that one cell to 2 cells to 4 to 8, right? As they're stacking up, right, which way they turn is determined by gene products, by proteins that were produced by the mom, right? So in that egg, they were already there. They're not being made, they've already been made, and they get used right away. So if mom, and notice he denotes male and female because of this maternal effect, right, this mom effect. If mom is dominant, all her children, because she makes that gene product, all her children, no matter their genotype, are all going to have that dextral turn to them. So notice whether they're male or female doesn't matter. This heterozygous genotype, although most of the time with dominant recessive, they would all be dextral, right? Anyways, right, because that's dominant and they're the heterozygous. For this one where she is the other way, right, all her children, even though they're heterozygous, if you mate with a dominant male for dextral, end up being the other way. Because again, it's mom's genotype that determines the phenotype. Not what the baby's genotype is, not what the dad's genotype is. It's what mom's genotype is. So when you cross 2 heterozygous individuals, you get this pattern of genotypes, but notice again, look at the phenotype. All dextral. Because mom is dextral. When you do the third cross, right, you get back some of the recessive. So let's look at this. So we would expect here at this point, right, if it was the standard Mendelian inheritance, we would expect 3 of them to be dominant and 1 of them to be recessive, right? We would expect 3 dextral and 1, however you say that, central. But it shows up in the 3rd generation, what we expect, right, for normal pattern of inheritance based on Mendel's laws. So later it was explained, right, as we said, dextral is dominant and central is recessive, but it is the mom that determines the phenotype, strictly just the mom's genotype. So when we look back at this, so notice heterozygous, right? Both the females here, do you see that? Heterozygous. So it doesn't matter what the dad is, all the moms have the dominant allele. So therefore, all the children are going to be dextral. Everyone's with me at this step? Okay. Because it's her genotype, not even her phenotype. Because notice here she's spinning the other way, right? But it's her genotype that is going to determine which way the babies rotate, which way the spin is for the shell. So then here, because we do have a big mixture of genotypes, when we do this cross, we get back that pattern that we expected in the first instance for the 2nd generation. And so how is it that we ended up with one that's spinning the other way? Who determines which way you spin? Mom. And so in this case, guess who that one's mom is? This one, because notice again, even though the mom is dextral, right, that's because her mother had the dominant gene here. But her herself, right, because we crossed 2 heterozygous, we ended up with the recessive. So now when she has babies, her babies are going to be the recessive. They're going to spin the other way. You see? So mom's genotype and phenotype don't even match. They are what their mom was. Ooh, have fun trying to predict those crosses, huh? Yeah, with maternal effect, yes. The mom's genotype determines the baby's phenotype. Mm-hmm. Yeah, it just depends on what you're mixing. You're going to get that switching. So let's see. And again, so notice mother's genotype, not her phenotype, because her genotype and phenotype, as you saw in that one of those, don't even match. Because her phenotype was determined by her mother's genotype. And as we said, and it doesn't matter what father is, doesn't matter what the embryo actually is. It matters what her mom was. That's maternal effect. So, of course, this is non-Mendelian pattern of inheritance. And so, as I said, this can be explained by the process of building that egg, that oogenesis. And so what they found is there are actually cells that surround the egg. They're called Ner cells, and these are diploid. Because remember, the egg itself is what? Haploid. It's haploid. It's half the information. So these surround the haploid egg. And so if the female is heterozygous, remember, she's going to produce both But she is going to produce the dominant allele. So you'll end up with dexteral spin to that shell and all the organs inside. The haploid oocyte receives, say, the D, the recessive allele. But again, remember, it doesn't matter because that oocyte does not make the proteins that are going to determine which way it spins. It's these nurse cells that are around the oocyte that actually produce the proteins. And since if the mom is heterozygous, she's going to produce the proteins to make that dexteral spin, right? Because she has at least one dominant allele. So even if she gives the little d to her baby, right? There's a little d in here and little d comes from dad. Doesn't matter. It's going to be dexteral because of that dominant allele that she has in her nurse cells that are nursing, basically preparing this egg for its development. Right, so it actually goes in to that egg. And so the egg doesn't make those, right? It relies on what's already been made by the nurse cells because it's going to rapidly divide once that sperm gets there and gives the other half of the DNA it needs for everything else. So I don't think we need to beat this one. There's a bunch of diagrams, just go over the same thing over and over again. We can skip over them. So maternal effects genes encode RNA and proteins that play important roles in the early stage of embryogenesis, right? And that's why they matter. So for cell division, cell cleavage, and in this case, the body axis orientation, the way it spins. And it's the accumulation of those products before even fertilization. So it doesn't matter what dad is, he's not even on scene yet. And it's not made from the babies, right, that haploid oocyte. Therefore, defective alleles in the maternal gene FX tend to have a dramatic effect on the phenotype of the individual. So in Drosophila, they've studied these types of maternal effect genes a lot. And there are several dozen of them that, like, if mom has defective, then the embryo won't form right. The larval and stuff, like their body plan, things won't line up like they're supposed to. And, you know, there'll be like those kind of crazy looking things you see in sci-fi movies, right? Or they just won't develop at all, right? Because what they needed, they didn't get, right? That information. And it doesn't matter what they have or what they got from dad, right? It was what mom had that determined it. And for her, it was what her mom had, right? Which is why she was able to be, but her babies can't be, because she's the combination of both her parents, which could have been a defective thing. But her mom was fine. Her mom had enough information that she could be. But the combination of what her mom gave and her dad gave, she doesn't have the right information to give to her babies. Still with me? That's a tough one. You have to kind of like go through it a couple of times, right? It's all about what mom gave you. Mm-hmm. Okay. No, you know, the thing is, oh, no, we're not on Module 2. We're still on Module 1. Yeah, this is the last of Module 1. Yeah. Which is why I was talking about the test and whatnot. Yeah.
So on maternal effect, these primarily like developmental?
Yeah, they're developmental. Yep. The developmental genes. Because again, they're made by mom for you to develop. So the you know, scary kind of crazy thing is if mom doesn't get the right stuff, she can't make good babies. It ends there. I'm kind of surprised I didn't put mutant flies on me. I don't want to freak people out. Have a picture of deformed looking flies. Okay, so the next is my favorite. It's epigenetic inheritance. And so we need to be able to know the similarities and differences on how dose compensation is achieved in different animal species. So in the case of mammals, right, X chromosomes, who has 2? Females, right? And males only have 1. So we have a dosage problem, right? Because we have 2, so we make twice as much as they make, right? Potentially, we could make twice as much stuff because we have 2 X's where they only have 1 X. So how do we fix that problem? So one of the ways is the X chromosome in mammals, one of them is inactivated. It's not used for females. So how could this affect our phenotype? So if we're not using one of our X's, will that change the way we look or the things that we can do? We just shut down one of those X's. No big deal? Both the X's are exactly the same? What do you think? No. Our X's differ, right? Because it depends on who you got it from, mom or dad, and what was on that, right? That they got from their parents, and so on and so forth. So yeah, it definitely can affect our phenotype, which X we're using. So this is known as epigenetic inheritance. And this refers to the pattern in which a modification occurs to nuclear genes, so we're still in the nucleus, or chromosome that alter gene expression. Right? So we're not going to use that information. This isn't a permanent thing. And so from generation to generation, it can change. It's basically individual picks. For women, for us, we pick which X we're going to use. This happens very early on in our development, though. Our cells have to choose which X they're going to use. And so, again, we're not changing sequence or anything like that. We're just not using one of them. So what do? We just modify the DNA or the chromosome. So they can occur as early on as oogenesis when the egg is forming, or spermatogenesis when the sperm is being formed, or early on in embryonic development, which is the case for us. For our female humans, early on in our embryonic development, we shut down one of our X's. So things that come into play with this is dose compensation and genetic imprinting. So what do we mean by dose compensation? We need to fix the problem that we don't have the same number of chromosomes in the 2 sexes, right? As we know, for males, they only have 1 X, we have 2. So you could potentially end up with too much stuff for females, or maybe not enough stuff for males, if we don't compensate for this difference. So of course, this has been extensively studied in fruit flies, as well as yeah, it can. Although the examples we're going to talk about are not disorders, but kind of cool looking. So depending on the species, this dose compensation happens different ways. So for us, right, we tend to focus on us, right? We have 2 X's, males have just 1 X. So what we do is one of our X chromosomes is shut down, right? We package it all up, really tightly bundle it, and we don't use it. In marsupial mammals, the parental-derived X chromosome is inactivated in all of its somatic cells of females. So what is parental? Paternal, paternal. Father. Right? So they have some way to know that that chromosome came from dad, that X, and we're not going to use it in females. They're just going to use the X they got from their mom. So that affects patterns of inheritance, right? Especially when we talk about X-linked characteristics. Right? Because if mom has an X-linked, say, disease, right, like hemophilia, or colorblind, I think might even be X-linked. And so mom has it, and if you're a female, dad can't rescue you, right? Because you're only going to use your mom's X. You shut down your dad's X. Does that make sense to you, Angela? So if you're a marsupial, it says that the X that you are going to use is the one you get from your mom. You're going to always shut down the one from your dad. So if mom's got something bad on that X, right, like colorblind or hemophilia, she's going to definitely give it to you in the case of marsupials here. So that's what they do. For us, we randomly pick between paternal and maternal.
There's no way to know if you're going to get it.
It depends on whether it gets picked or not and in what cells. Right? So in the case of color blindness, right, you have a 50-50 shot maybe if you have a defective and a good, right? Where if you have 2 defectives, clearly you're going to be colorblind. Doesn't matter because both of them are defective. You see? But if you're heterozygous, then it's going to be like a 50-50 chance. It depends on which one you turn on and which one you turn off.
Oh, what's
Drosophila, those are fruit flies, right? So for these guys, the X chromosomes in males is increased 2-fold. So they don't do like us. They don't shut down one of their X's in the females. Instead, the male upregulates expression of his X chromosome. So females don't shut anything down, and the males just make more of the stuff from their X. Now this next organism, I want to say it's a worm. Should have looked up on what it was. It is a worm. Yeah, I was pretty sure it was a worm. It's a little wormy, guys. So the interesting thing about worms is the XX, or what we would commonly refer to as a female, is actually a hermaphrodite. So they're able to reproduce on their own. And so what we would consider males only end up with 1 X. And the level of expression of both X chromosomes in hermaphrodites is decreased. So the fact that she's got, or it, I don't know what you call a hermaphrodite, it has 2 X's, to compensate for that, it downregulates and only makes half of the information, half of the gene products that it normally would. So as you can see, there's lots of variation, right? We're going to focus, of course, on what we do, what mammals do. Placental mammals as opposed to marsupial mammals at that.
When it downregulates, is it because the cropping process, the replication process, they recognize the...
Yeah, so it has a way of detecting like how much it's made of something. So it doesn't make as much because it doesn't need to, because it's got 2 X's it can use. So it doesn't need them to use them to the full extent, use that information to the full extent. So you have 2 working copy machines, right? Spread out the work and do half as much, right? With 2 copy machines as opposed to just 1, right? So you got to find a way to deal with this problem of having too much or too little, right? So for us, for females having too much X chromosome, we just shut one down, right? And we just use one of them. For marsupials, they're going to shut down one of the females, but they're going to be very specific in that, right? They're going to shut down the X that they got from their dad. But that creates a problem, right, as far as like inheritance, right, if mom gives you crappy stuff. For fruit flies, the guys just work harder, right? They produce more product from just one copy machine, from one X. For these guys, that have the double X, they just produce less. Right? They don't fully express 100%. Still with me? All right. Okay, so it's not well understood in some other species, right? So those are ones that we have pretty good understanding, but birds and fish, not so much. In birds, the sex chromosomes, the really large sex chromosome is known as Z, and the other one is denoted, that's smaller, as a W. Not that they look like a Z or W, quite honestly, I don't think. Those are the letters that they picked for them. I don't know why. I don't know the background on that. But unlike for us, it's males that have the two large chromosomes and the females that have the large and the small. The non-matching sex chromosomes. So that gets confusing right there for us, right? Because you're like, oh, it's opposite than what we're used to, right? It appears that the Z chromosome in males does not undergo, it doesn't condense like we do, right? They don't condense it in any way, that extra Z they have. They believe the variation in gene expression of some of the Z-linked genes in males and females so there's a variation in expression. So they either upregulate or downregulate different genes for different reasons, right? So there's no clear-cut answer for all the genes. They're not compressing one chromosome down like we do. But they're definitely regulating the different genes at different rates. Does that make sense to you guys? Okay. They may lack a general mechanism, but some compensation may happen for specific genes, right? So specific ones they're purposely making more of if they're the female from the Z, or if they're the male, they may be making less of it. So Barr, and the other guy that tends to get forgotten about because he didn't get to have the name associated with the structure. Bentram. What is it? Ewart? What a name. Murray Barr and Ewart Bentram. Bertram. So these guys identified a highly condensed structure in the interphase nuclei of somatic cells of female cats, but not in males. So they're looking at, our regular cells, not our sex cells, and they're noticing that at that stage of development, right, we're not talking about when they condense the chromosome or anything like that. We're not talking about prophase or metaphase when they line up, and anaphase and telophase when they're coming apart, right? We're talking about when they're not even condensed. They found a region within the nucleus that was condensed, right? It's DNA, it's protein, it's condensed. Why is it condensed? And they only found it in the females and not in the males. So what's the major difference between males versus females? Right, we have an extra X. We have two X's. They have an X and a Y. So this structure is now known today as the Barr body, but what it actually is one of the X chromosomes condensed and inactivated and not used by the somatic cell. So other scientists, continued on that work, right?
So it's just a super tiny armed X.
No, I mean, the DNA, the thing is, the X only happens when we've copied it, right? When you're at prophase. And then you're splitting because you're the two sister chromatids and you're splitting them apart. So normally it's just one big long chromosome, right? And the DNA is wrapped around histones and stuff, but they complex and uncomplex and you know what? I posted a link for you guys, and I think I'm gonna play it. Because it will explain it. So one of the professors who used to be here, but now she's in med school in the Caribbean, she posted this actually on her Facebook page. Recently, she's studying this, and I was like, oh my God, this is perfect timing for our class. So I have it under our Module 1, which yes, we're still in Module 1, but we're at the end. And there's our Chapter 5, and here is Stripy Women. Yep, it's on IFL Science. So why are women stripy? Well, because we shut down one of our X's. And this shows it. Let me see if I can, how do I make it big? Will it let me make it big? There it is. And of course they're going to have a commercial. There we go.
Made up of 6 billion letters of genetic code. Now, your DNA is split into 46 pieces, each 3 to 4 centimeters long, called chromosomes. Now, normally we think of chromosomes as looking like this, but they only take that form when a cell is ready to divide. So usually DNA is just a wiggly thread within the nucleus. Now, if you can imagine, DNA is only about 2 nanometers wide, but a chromosome is centimeters long. So you would think that it would get tangled worse than the headphones in your bag. So the DNA is actually wrapped around proteins called histones. Now those histones have wiggly tails, which will come in handy, as we'll see in a moment. Your unique set of DNA first formed when 23 chromosomes from your mom mixed with 23 from your dad. Now 22 of those chromosomes from each parent form matching pairs, but the 23rd set is the sex chromosomes. So 2 X chromosomes make you female, and an X and a Y make you male. Now since the male sex chromosomes are different, both can remain active for the rest of your life. But for females, one of the X chromosomes needs to be inactivated in order for proper development to occur. This happens when a female embryo is just 4 days old and consists of only 100 cells. Right now, in this cell, the X chromosome from dad and the one from mom are both active. But through a tiny molecular battle, one of the X chromosomes wins and remains active, while the other is inactivated. This is done by packing the DNA closer together and making modifications to those dangly histone tails that signal this inactivation. New structural proteins are also added to bind everything closer together. And finally, methyl groups, these tiny little molecular markers, are added to the DNA to basically signal to the cell that this DNA shouldn't be read. So all of this together makes the DNA very difficult to access for the molecular machinery that would harness the code in this DNA. It is switched off. This DNA is silenced. In contrast, the active X chromosome DNA is more spread out. This allows better access to the genes on the chromosome. Histones can be slid along the DNA or removed entirely. And histone tails have a different modification, signaling this DNA is active. Now, all of this makes it possible for RNA polymerase to access and transcribe this DNA into messenger RNA, which then goes out into the cell and is used to make a protein. Now, what's surprising about X chromosome inactivation is that it seems kind of random which X chromosome wins. I mean, in some cells, dad's X chromosome wins, and in others, mom's X chromosome wins. So this 100-cell embryo ends up with a mixture of active X chromosomes. But from this point forward, as these cells divide, they maintain the active X chromosome that they had inside. So all of the cells with dad's active X chromosome give rise to further cells with dad's active X chromosome. And this continues on into adulthood. So if you could look at a woman's skin and see which X chromosome has been inactivated, you would see a stripy pattern, which shows the growth and migration of all of these first 100 cells when the embryo was just 4 days old. Now, of course, you can't actually see that in humans, but you can see this with calico cats. And that's because the gene for coat color is actually on the X chromosome. So just by looking at the pattern of her spots here, her dark and light spots, you can see where her mom or dad's X chromosome has been inactivated. And this also shows us that only female cats can be calico cats. And that's because, well, only female cats can inherit 2 X chromosomes with 2 different color genes. Now, this is just one really cool example of epigenetics your genes. For example, it's epigenetics which makes a pancreatic cell capable of producing insulin, because that gene is switched on there, but switched off.
I want to pause that one or close that one because he's starting to go into stuff where I don't even know if we're going to get to. But you can listen to it on your own. So that I think that was really good because it had the animation along with it, right? That was like perfect timing. Thank you, Miss Adams. Does that help? Right? In the understanding. So the cool thing with the calico cat is that the orange or the black is on the X chromosomes. So if it's black, whatever X chromosome was turned on was the black one in that region. And if it's orange in that region, that one was turned on, and the other one, the black one, was inactivated, became the bar body. So can you have a male calico cat? Yeah. But it's a sterile male because, unfortunately, he got 2 X's and 1 Y. Right? So those are really rare. It is possible, right, though, it is possible for a male cat to be calico, right? But he won't be able to reproduce because he has 2 X's. That's how he's able to have that calico color. And also has a Y, which is why he ended up with a penis.
Which chromosome is the trisomy? Is it 23? So it is.
No, it's trisomy 21. It's 21. For the Down syndrome caused by trisomy 21. It's the 21st chromosome that you end up with 3 of them. And that creates the condition known as one of the Down syndromes. There's actually more than one Down syndrome, but that's the most common Down syndrome. And we will get to that one. Okay.
So is it randomly spread throughout the body?
So what happens is when we are 100 cells, right, they choose between one X and the other, and they shut it down. And even when those cells divide, whatever one is shut down, they copy it and everything, but they keep it shut down. That particular one that was picked to be shut down is shut down. So as he said, if we were kind of like a calico cat, right, you would be able to see that pattern on us of what cells arose from what cells. And it would almost create like those stripes like you see on the calico cat. Because, again, that imprinting that happens of choosing which one it's going to use happens early on. So all cells next to it, right, are going to either be black or orange depending on what cell they arose from for a calico cat.
So if we're looking at like X-linked inheritance, like for disease, would it be possible then for a female to not just be a carrier but actually have the disease if she predominantly?
Yes. So that's what Aaron and I were kind of talking about earlier, or Angela, they were like, well, how does that affect diseases, right, that may be on the X chromosome? It depends. Like if you're heterozygous, that means that you have a defective and a good one, right? But then you're only going to use one of those. So it really depends on which one you shut down and which one you don't. And in the case of like maybe color blindness, right? If you pick the right one in your eyes, you'll be fine. If you pick the wrong one, you're going to be colorblind, right? And you have no control over that. They believe for the most part it's pretty random, although So there's our cute little Callie, right? And that's because we shut down one of our X's and we condense it so much that it forms this very characteristic structure even within the female somatic cells, right? That we know as the Barr body, because Barr helped discover it. So, and of course, the orange and the black is X-linked. I'm trying to decide if I want to go over, if I want to skip some of this. I'm trying to remember where it's at. Okay, here they start talking about it.
Is this the same bar as Epstein bar?
No, different bar. Different bar. Okay, so at the molecular level,
So we're skipping all the other stuff?
for right now, it gives you another example, right, that we can do in our examples if we need to. So at the molecular level, right, on our X chromosomes, there are genes, right, within these regions. So they give them these abbreviations, right? So one of them stands for X inactivation center, right? So this is the section of the chromosome where the inactivation is going to happen or not happen. So from this DNA, so this is representing DNA, we make two types of RNA, right? And of course these are acronyms. Where is what they stand for? Here it is, right? So XIST stands for X inactivation specific transcript. So this RNA is only expressed on the inactive X chromosome. It doesn't encode a protein. It just makes this RNA. But what this RNA does is it actually interacts with the chromosome, the X chromosome that it inactivates. And then other proteins, right, as he said, come together, right, and really condense that chromosome, that inactivated one. So we're not going to go into all the particulars of that. What else did I want to say? Okay, what's the other one? So the opposite of that, you see this one, prevents it from being compacted. So this one is going to cause compaction. This one prevents compaction. Now, I don't think they've worked out exactly why one binds to one chromosome and not the other. And like I said, from what we've studied, it doesn't, I mean, especially with the calico cat, when you look at the calico cat, right? She doesn't shut down all the ones she got from her dad or her mom, you know, kind of like the marsupials shut down all the X's from their dads. It's random. It appears to be random anyways. But there is a molecular basis behind it. I mean, we do make these transcripts. They do attach to one chromosome versus the other. Shut down one and not the other. So, wow, that's two of them. Let's see if we can get through the third one. And then we can leave extracellular to Thursday. So the next is genomic imprinting. So this is where DNA is marked, and that effect is maintained throughout the life of the organism that inherits that marked DNA. So this is almost very similar to what we just talked about, right? Where We figure out who's going to be the inactivated chromosome, and that continues through the woman's entire life, right? We don't ever unactivate the one we activate. We condense. It's shut down. So as you saw in the animation too, like they add sequences, they add methyl groups and such, right? They modify it so that they know, right? So we're imprinting, we're marking it. These still follow Mendel's pattern of inheritance, the genes themselves. But it depends on how the genes are marked. So it basically depends on, okay, are we going to mark moms and you're going to inherit moms, maternal, or we're going to do paternal? Or I should say, not whether you're going to inherit, whether you're going to express which one, the one you got from mom or the one you got from dad. Not both, right? It's a choice. One or the other. So this is termed as monoallelic expression. So even though maybe you have two alleles, right? One for mom and one for dad. So imagine you had AB blood type, right? But instead, we're going to shut down whatever you got from mom all the time. Or we're going to shut down all the time what you got from dad. In that case, your blood type would be either A or B, right? Even though you have both alleles. You only use one of them. Still with me? Okay. This example is not too hard to follow. Okay, so this gene codes for a growth hormone. It's called insulin-like growth factor 2. So that's why it's IGF, right? So insulin growth factor number 2. So if you have this normal functioning gene, right, it's a growth hormone, so it's going to determine your growth, right? So if you're going to be normal sized or not. So if your gene is working, you got the non-mutant copy and it's A-OK, then you should be a normal sized mouse. Imprinting results in the expression of a paternal but not a maternal allele. So we're going to mark, right, them, and we're only going to use the paternal. So whatever chromosome, right, we got from dad for this trait is what we're going to use. The maternal is shut down, right? It's not utilized. So it doesn't really matter what mom gave you because you're not going to use it anyways. But, of course, it may determine what your babies are and their babies. So minus, of course, is a loss of function, right? You don't produce this growth hormone, and therefore you're going to be abnormal size. You're going to be smaller than normal, or what we call dwarf. Okay, so mom is a little baby, right? Because notice she's got both defective genes. Or maybe. But that's her genotype. Dad's genotype is he's normal. So they mate. But remember, we're going to mark moms versus dads. Moms, notice she's the minus, are always going to be silenced. We're going to shut moms off. We're going to only use what we get from dad, which is great. These guys won, right? Because they were going to get something defective from mom. These guys are going to be completely normal because they're only going to use the normal gene they got from dad. But if you do a reciprocal cross, you do the flip of that, where dad is abnormal genotype, right? Mom's genotype is normal, but remember, we're going to silence moms, hence the red, right, triangle. Dad's is what we're going to use, but unfortunately Dad's is defective. So you're going to end up with dwarfs. Right? And so this is the comparison, right? Here's normal size myces. There's the little baby dwarfs. So notice both offspring are heterozygous, right? But it isn't what their genotype, it is what their genotype it is, but more importantly, it's what their genotype is that they got from their dad. It's going to determine what they are. So it's being imprinted, right? It's being marked as to which one is being inactivated and which one's being activated. In this case, we're inactivating whatever comes from mom. And we're only using what comes from dad. Reciprocal means the opposite. You guys know that, right? Okay. You'd be surprised. So at the cellular level, imprinting is epigenetic, right? We're marking stuff. And so the first is the establishment of the imprinting. So during gametogenesis, right, when the gamete forms, it's determined. And then it's maintained throughout the development and in the adult. But it's going to be basically erased and reestablished in the germ cells. So let's look at lineage here. So it's been established, right? This sperm is marked, active, and the egg is inactive. So then they join up, right? And so, of course, because this was normal, he's normal, right? And because he got normal from his dad, he's normal, although moms are defective, right? Then when she sets up hers, right, she's going to give half of her kids will get the normal that she got from her dad, and the other half are going to get the defective that she got from her mom, right? You see that? But because she's a female, she's going to silence all of her stuff. Doesn't really matter whether she gives defective or not to her babies, because she's female. All of her stuff gets marked for silencing. Where on the flip side over here with the male, he's heterozygous, right? So he can give either defective or good genes to his babies, and all of his are turned on. So it really depends on which sperm he gives his babies, which will determine whether they're going to be normal sized or not. Did y'all see that? The fastest swimmer will have the defective gene. I don't know about that. I don't know if swimming is affected. But yeah, it's all about who gets there first, right? All right, so I think we will stop there for today. I think, again, they just give another example. But we'll come back to this one before we move on to the next. Yeah, they just talk about how it's done at the molecular level. Basically, methylation. Right, we're just going to label it with methyl groups. So pick up where we left off. If you haven't, for some reason, done your Chapter 4 problems in Connect, get those done. We're going to finish this up. Right, on Thursday, we'll work through problems. So if there's problems in the book that you particularly want me to go over, bring with you, right? Or write down, or even email me in advance so I can prep it. But I can do on the fly too. But we can get more done if I know ahead of time, right, what ones you're struggling with. If there's any particular ones from that homework, which is why you need to get it done ASAP, you want me to go over. Test is either going to be Tuesday or Thursday next week. I will definitely know by Thursday when we're actually going to do it. If for some reason we don't have it on Tuesday, we have it on Thursday instead, we will cover new material on Tuesday. You can't waste a day. That won't be on Thursday's test, right? It'll be new for the next. Any questions? Okay, all right. So work on problems so we can go over the stuff that's getting you.